Uh, Hello, everyone. My name is Kauthar Omar, and I'm going to take you through my master's thesis, which I did in Pwani University, Kenya, in collaboration with the Center of Genomic Regulation on quantifying allo-coprophagy in heterogeneous stock rats from deep shotgun sequencing data of the gut. Microbial exchanges can occur in a variety of ways, and this influence the microbiome composition, and this can be through touching of similar surfaces, and also through contamination of water sources which the microbe find the way to our households. And we see in a study by Dill McFarlane that shows that, and Raulo et al., that shows that social partners share a similar microbiome. And in the Dill McFarlane study, we see that a couple of human adults shares a more similar microbiome than adult siblings. And also in the Raulo et al. Study, we see that mice of the same social network share a similar microbiome. And in the paper, the person-to-person transmission landscape paper, it actually shows the transmission dynamics of microbes. And all this is important to study as microbes affect many traits and diseases, such as inflammatory bowel syndrome, among others. In this study, we modeled the transfer of commensal microbes through a process in mice called allo-coprophagy, where we use the HS model rats. And allo-coprophagy permits the transfer of microbiome between rats sharing a cage. And allo-coprophagy is when one rat consumes the feces of another rat, and in our case, where a focal rat consumes the feces of its cage mate. This process is difficult to quantify through direct observation and video recording, as it occurs several times a day. Therefore, the quantification would require observing cages for many hours, which is very time consuming. So in my study, my objective is to detect and quantify allo-coprophagy in heterogeneous stock rats from deep shotgun sequencing data of the gut. And my hypothesis is and approach is when I sequence, when allo-coprophagy takes place in the feces of its cage mates, we'll have cells from its epithelial will have cells from its epithelium. So when we sequence the cecum of the focal rat, we expect to find cells from the focal rat, cells from its cage mate, and microbial cells. And once sequenced, reads from all the three, and we will select the rat reads by aligning it to the rat reference genome. And we are not going to use the microbial reads in our case. So my data set comprises of 32 heterogeneous stock rats from Lea Solberg of Forest University. These rats were co-housed in pairs for 5 weeks, where we had some that were fasted and some that were not fasted. And for the deep shotgun metagenomic reads, we have sickle samples, which are the sickle whole, targeting the whole, sickle squeezed, sickle leftover, and fecal. And we had 150 paired end reads targeting 50 million reads. So my approach or method is I had my gut samples, which are both sickle and fecal. And for these 32 rats, I had 95 samples, which I genotyped. I received genotypes for the 32 rats from the collaborators. And once I selected my rat reads, I did the calculations of quantifying the sample mixtures in a package called MB Mixtures, which uses both the metagenomic data and the genotypes. And I compared the sample mixtures, and if I found the mixtures in cage mates greater than non-cage mates, it would be a case of allo-coprophagy, allo-grooming, experimental contamination, and noise. And if not, this would be a case of experimental contamination and noise. For the quantifying of sample mixtures, I mainly used the tool MB mixture, which was used by this paper by Lobo et al. To identify sample mix-up and mixtures. But for my case, I identify sample mix-ups and also sample mixtures which I suspect to be allo-coprophagy. So the key plots that were generated by the tool MB mixture and that I used in my analysis were the sample mix-up or sample swap plots, which in this case, in the x-axis, we have the rat genotypes, which are the 32 rats in my 32 experimental rats. And on the y-axis, we have distance from genotype, which is the mismatch between a gut sample and each of the genotype. And in this case, sample X, this is a case of a sample mix-up or mislabel or swap, which we have another genotype having less distance from the genotype than the actual label, which is sample X. And also in the same case of sample mix-up or swaps, I'm going to look at another plot, which is plot B, which has the self-self distance against the best distance against the self-self distance. So the self-self distance is the distance between a sample and its genotype, while the best distance is the minimum distance seen between the gut sample and any other of the 32 genotypes that are considered in this study. So these samples that we see in this quadrant are actually we have a greater self-self distance than best distance. Therefore, they are likely to be cases of sample swaps, mix-up, or mislabels. I'll be using these three terms interchangeably. So the next plot that was generated by the package, and I also use in my analysis, is the sample mixture plot, which has the LRT statistics, likelihood ratio test statistics, against P. P is the proportion of contamination or the proportion of the sample coming from any of the 31 rat, excluding itself. So for me to identify a sample mixture, I am considering P and the LRT, where this, like for example, Z is a case of a sample mixture between sample X and sample Z, because Z has a high proportion of 0.176 coming from sample Z, and also the LRT is high. So for the sample swaps, or which was a swap, I identified 5 sample swaps, which was a swap between 5 samples. And when we look at the distance plots, we actually see, for example, in this sample 011, sample 011, we see it has it has a smaller distance from the genotype with sample 027 than its own genotype, which is 011. Therefore, we suspect that the true identity of 011 is 027 and not 011. The same thing to 027. We see it has a smaller distance from the genotype with 019 than its own identity 027. Therefore, we suspect the true identity or label of 027 is from sample 019. And this happens in this pattern that I have drawn the arrows, as you can see in this image. And when we look at at the sequencing plate and we try to look at what happened, we see all these 5 samples that had a sample swap were actually in a linear manner. So we suspect that in the processing plate, what happened is 011 was omitted, and therefore when we correct, and therefore it took the position of the previous sample and it was lastly added, because after the sample correction, this is what we see. So when we look at the best distance against the self distance plot, before the correction, we actually see these samples are in this quadrant. That means they have a greater distance with its genotype than the best distance. Therefore, these are the same 5 samples we saw here, which were a case of sample swaps or mislabels. And after correcting according to this pattern that we used here, we actually see all of them being in the diagonal line. Therefore, their self-distance is same, their self-distance and best distance is the same, or the difference is small. So I'm going to show examples of high levels of sample mixture. And in this case, this was an example of high sample mixtures, which was between non-cage mates. That means in this case, it's a sample mixture between 0.25 and 0.27, and it is very, very high. As you can see, the P is 0.55. And this is a case of contamination. As you can see in the gut sample processing plate, we actually see 0.27 located next to it. And also in the spleen sample processing plate, we also see 0.27 located next to 0.25. And when we see sample mixtures between cage mates, I'm going to show an example, which is the example of rat 032, where it was a mixture of 032 and its mate 031, where P was 0.16. This is likely not a case of contamination, as we can see in the gut sample processing plate. 031 was not in the wells. And when we also look at this, so when I looked across all the 32 samples, I looked at the proportion of contamination from mixtures of cage mates and mixtures of non-cage mates. We actually see the mixtures of cage mates is slightly higher, although this is not statistically significant, and even from the box plot, the difference isn't significant. So from this, I was able to identify 5 cases of sample mislabels, and I also identified a case of suspected allo-coprophagy, which was sample 031. However, this did not have statistical support across the experimental data set, and I suspect mainly this is due to the small sample size I had of 32 pair housed rats. 16 cages is not enough to detect a subtle process like allo-coprophagy. And for my future prospects, I'm considering using 2,000 co-housed rats, which they are co-housed in pairs and groups of 3, which are available at the Baud lab. And I'm also going to investigate microbial transfers by looking at the bacterial strain sharing. I'd like to acknowledge all the supervisors I had for my thesis, Baud Lab, Pwani University, and the partner institutions. Thank you so much.